Today I'll be presenting the uptake of the anticancer drug Cisplatin mediated by the copper transporter CTR1 in yeast and mammals. This paper was created by Ishida S., Lee J., Theo DJ, and Hershkowitz. So, looking at the mechanism of cisplatin, we don't exactly know how the cisplatin enters the cell. However, once the cisplatin enters the cytoplasm, chlorine on cisplatin exchanges with water, which activates the cisplatin, and we can see, and based on the research, we can see that DNA is a critical target for cisplatin cytotoxicity, as it is evident looking at the hypersensitivity to cisplatin of both prokaryotic and eukaryotic cells deficient in DNA repair. The most prevalent cisplatin-induced DNA adduct is an intrastem cosine, in which the platinum covalent bound to the N7 positions of the imidazole ring of two adjacent guanines. The intrastrand cross-links are replaced by the nucleotide excision repair pathway. And another thing we need to know before we dive into the research, it is the sensitivity to cisplatin. There are several methods to achieve it. First, there's the overexpression of multi-drug resistant protein MRP2 or copper transporting P-type ATPase ATP7B, which results in the increased resistance and decreased cellular accumulation of cisplatin. Genes that alter intracellular levels of files, which are highly nucleophilic and can react the intracellular cisplatin, have been demonstrated to affect levels of resistance to cisplatin by sequestering intracellular cisplatin. Mouse cell deleted for MSH2, a mismatch repair protein, exhibited increased cisplatin resistance. It is hypothesized that mismatch repair proteins detect the cisplatin-induced damage and initiate a series of events that result in cell cycle arrest and death. Saccharomyces cerevisiae biochemical and genetic screens have identified several genes that govern cisplatin resistance. The mechanism is unknown. The purpose of this paper is to describe the identification of the CTR1 gene as a major determinant governing cisplatin resistance and accumulation in yeast and mice. So when studying the yeast of CTR1 in yeast, first, the researchers created the isogenic strains carrying deletions. Then they selected for the cisplatin-resistant mutants, and then by exposing yeast cells to different concentrations of cisplatin, they were able to select mutant yeast cells that were resistant to cisplatin, and they were prepared. And the cells were prepared for platinum measurement, in which the cells were washed, lysed, and centrifuged. And later, the prepared cells underwent atomic absorption spectroscopy, which was used to measure platinum. And now focusing on the studying of CTR1 genes in mice cells cultures were cultured, and then later mouse cells were treated with 64 copper in the presence of 100 molarity or 100 micromolarity cisplatin or 10 molarity unlabeled copper sulfate for 2 hours. And moving on to the results. The cisplatin resistance of yeast mutants. The mutants that showed the highest degree of resistance was defective in the MAC1 gene. Deletion of the complete MAC1 or F resulted in a 2.5-fold increase in cisplatin resistance relative to wild-type cells. The number of cells that survived a 2-hour treatment with 1 micromolarity cisplatin was 1,000 times greater in the MAC1 mutant than in wild-type cells. MAC1 encodes a protein that activates transcription of catalyzed genes and genes involved in uptake of copper and iron. To address whether increased cisplatin resistance of the MAC1 mutant is due to the inability to transcribe MACP1 target gene, researchers deleted each of its known target genes, namely CTR1, FRE1, FRE7, CTR1, and CTT1, and measured the sensitivity of the mutants to cisplatin. Of the 5 mutants, only the CTR1 strain showed a level of resistance similar to the MAC1 mutant, which can be shown in Figure 1a. These observations suggest that CTR1 is the major target for MAC1P with respect to cisplatin resistance. CTR1 encodes a high-affinity copper transporter. Cells deleted for CTR1 are resistant to growth inhibition caused by high levels of copper in the medium, fail to grow on low copper medium, and are defective in enzymes such as superoxide dismutase and in cellular activities such as iron uptake and respiration that require copper. To determine whether this cisplatin resistance of CTR1 mutants result from an effect on intracellular proteins that mediate intracellular copper trafficking and neutralization, we measure cisplatin resistance of ATX1, CCCC2, FET3, Lys7, SOD1, COX-17, and SCO1 mutants. Significant increases in cisplatin resistance as compared with the CTR1 mutant in quantitative assays were not observed. Thus, defects in proteins involved in intracellular copper distribution or utilization apparently do not play a major role in cisplatin resistance in yeast. The researchers also tested for involvement of other yeast copper transporters in cisplatin sensitivity. Another high-affinity transporter, CTR3P, and 2 low-affinity transporters, CTR2P and FET4P. Deletion of CTR2 and FET4 did not affect cisplatin resistance. Deletion of CTR3 resulted in a slight increase in cisplatin resistance. Although the CTR1 and CTR3 proteins have redundant roles in copper uptake, they have distinct structural features that could be responsible for a difference in their function and cisplatin resistance. These observations indicate that of the yeast copper transporters, CTR1P plays the most prominent role in governing cisplatin resistance. Now, looking at the effects of CTR1 in mice, mammals contain functional homologs of the yeast CTR1 gene, HCTR1 in humans, and MCTR1 in mice, which complements the yeast CTR1 mutant for intracellular copper deficiency. To test for a potential role of mammalian CTR1 in cisplatin resistance, the researchers prepared mouse cell lines from 2 dependent embryos that were wild-type heterozygous or homozygous for a knockout allele of MCTR1, and tested their cisplatin sensitivity. As shown in Figure 5a, homozygous mutant cells were 8-fold more resistant than wild-type cells to cisplatin, and heterozygous cells were 4-fold more resistant than wild-type cells to cisplatin. Similar results for survival or uptake using an independent pair of wild-type and homozygous mutant cell lines. The levels of cisplatin accumulation were also reduced accordingly. Heterozygous cells exhibit a 35% reduction in cisplatin accumulation compared with wild-type cells. Homozygous mutants exhibit a 70% decrease. A similar greater reduction in copper uptake was also observed with the wild-type, heterozygous, and homozygous mutant cell lines. These observations suggest that MTCR1 protein functions as a cisplatin transporter in mice cells and that its human homologous HCTR1, which is 90% identical MCTR1, may mediate cisplatin uptake as well.